Hello, my name is Hassan Salhab. I'm a PhD student at Kingston University. Today I will be delivering a presentation about high-performance liquid chromatography, an important technique for the analysis of pollution and its removal in wastewater effluent in Thailand. Today's session is about background related to the occurrence of pharmaceutics, hormones, and pesticides in fresh water and wastewater in Thailand, principle of high-performance liquid chromatography technique, state the advantages and disadvantages of this technique, and examples of using HPLC in environmental analysis, and the analysis of water pollution using this technique. Background: pharmaceutics, including female hormones, can enter the environment from different routes, from hospital, from industrial, and from other sources, human sources. And pharmaceutics can be detected in the environment in wastewater and surface and groundwater at a concentration range between nanogram per liter and microgram per liter. Occurrence of pharmaceutics in surface and groundwater in Thailand. A recent publication has shown that there is 4 main female hormones that it's been detected in surface and groundwater in Thailand. 3 of them are natural female hormones, E1, E2, and E3, and a 4th one is a synthetic female hormones named as EE2. Estrogenic substances such as natural estrogens such as E1 estrogen, 17-beta estradiol, E2 estriol, E3, and synthetic hormone 17-alpha ethinyl estradiol, EE2, can enter the environment from many sources, including hospitals, industries. A low concentration of estrogenic substances can cause harm to the environment system, such as fish, reptiles, birds, and even humans. Table 1 summarizes the concentration of estrogenic hormones in wastewater in Thailand. And it's been expressed as detected as microgram per liter. Principle of HPLC. Figure 2 represent a common configuration of HPLC UV. This picture has been taken from the analytical lab at Kingston University. The first orange arrow represents the mobile phase, the solvents. The second arrow is the injector. Third part of HPLC is HPLC column. The fourth part of HPLC instrument is HPLC pump, and the fifth part is the UV detector, and the sixth part is the data analysis. Principle of HPLC. HPLC stands for high performance liquid chromatography. It's used to separate compounds mixed in a solution based on the distribution between the stationary phase and the mobile phase. The common used stationary phase in HPLC is mostly silica gel, as shown in figure 3, and there is other a common stationary phase, including the fused core particles or multimodal stationary phase, as shown in figure 4. Mobile phase delivery system. Mobile phase can travel through the column and can be temperature controlled. And the mobile phase made up of organic solvents such as methanol or acetyl nitride, and it can be aqueous phase such as water, and pH can be adjusted when necessary. It's important for the degassing in order to prevent bubbles that could affect the retention time of the components. And as you've seen in the picture, the direction of the mobile phase goes all the way from the mobile phase through HPLC parts and finally end up into the waste bottle. Sample injection port. Samples are injected using a microsyringe into the injection port. This is done with a sharp plus of the sample. It's important to have a narrow chromatographic peaks which gives a greater separation of the substance and increased its sensitivity. Typical injection volume used is 5 to 20 microliter. Gaussian's peaks are the best chromatographic peaks, and samples in a solvent similar to a mobile phase composition at the start of the run will lead to the best peak shape. Figure 7 represents a picture taken from analytical lab at Kingston University. Is the injection valve of the Agilent Hplc instrument. Stationary phase, there are 5 different types of chromatography. Normal phase chromatography, the stationary phase is polar, while the mobile phase is non-polar. Reverse phase chromatography, the stationary phase is non-polar and the mobile phase is polar. Size exclusion chromatography is made up of a polymeric beads, for example, polystyrene. Ion exchange chromatography, the stationary phase is made up of resins and the mobile phase is made up of saline solutions. It's used for the separation of biological molecules such as proteins, amino acids, and nucleotides. Affinity chromatography, it consists of specific ligands, for example, antibodies coupled on beads. So in our project, we need to analyze, detect the pharmaceutics in ground and wastewater in Thailand using a reverse phase C18 column stationary phase, the most used stationary phase. Analytical column, the typical analytical column used in HPLC, its dimension ranges between 5 to 25 centimeter long and 1.2 to 5.6 millimeter in diameter. The commonly used stationary phase, silica derivatized with C8 and C18. The difference between C8 and C18 is the number of hydrocarbons inside the column. The particle size is between 2.6 and 5 micrometer, porous or fused core, and it's important to have a guard column at the both ends of the column in order to protect the main column from damage. In this slide, we will compare a very advanced technique called ultra-high performance liquid chromatography, UHPLC, with HPLC. As you see in figure 12, there is a comparison between HPLC chromatogram and UPLC chromatogram. HPLC technique is an advanced technique that operates a high pressure between 6,000 to 15,000 psi compared to HPLC. UHPLC achieves greater resolution compared to HPLC. Separation in UHPLC are faster compared to HPLC. The optimum column particle size used for UHPLC is between 1.7 to 5 micrometer. UHPLC can be used in different applications. In medical and pharmaceuticals, analysis of urine and drug stability. In food and forensic, analysis of sugars and drugs in blood and urine, and it's used for environmental purposes like detection of pollutants in wastewater influence. Detectors. This figure 13 represents a picture taken from the analytical lab at Kingston University, Agilent UV detector. There are 2 types of detectors, solvent property detector and solute property detector. Solvent property detector, the purpose of this detector is to change the mobile phase, detects the change of mobile phase properties. While solute property detector detects the property of the solute. There are different types of solvent property detector, including conductivity detector and light scattering detector. Light scattering detector is used for the characterization of antibodies and protein. Solute property detector. There are many types of solute property detector, including UV visible detector, photodiode array detector, fluorescence detector, electrochemical detector, and mass spectrometry detector. Once compounds have been detected, so it will generate a chromatogram. So data analysis is important in HPLC. We have a new scientific term called N. Capital N is the number of theoretical plates per column, equal to 5.54 TR over W at half height square. So TR is the retention time of the peak, W 0.5 is the width of the peak at half height. In order to determine the resolution between two peaks on HPLC chromatogram, there is a rule, resolution equal retention time of the first peak minus retention time of the second peak divided by 0.5, the width of the first peak plus the width of the second peak. Now we will be talking about the advantages and disadvantages of HPLC. One of the advantages of HPLC, it is high sensitive and it's reproducible technique, and it is a high selective technique, and the quantitative sample recovery and the high speed compared to other techniques, usually the runtime between 2 to 30 minutes. Limitation, one of the disadvantages is that it's less separation efficiency than gas chromatography, and it produces waste solvents. Applications of HPLC. HPLC can be used in many applications, including environmental application, such as monitoring of pollutants, a detection of phenols in drinking water, for example. It's been used in pharmaceutical applications, such as study of drug stability and tablet dissolution, and it's been used in medical and clinical applications, such as the analysis of urine, antibiotics in blood, and it's been used in forensic application, analysis of drugs in blood and urine, and it's used further in food analysis, such as the analysis of sugar level in food products. And it's finally been used in biochemical applications, separation of inhibitors and substrates, and this has been used in drug-drug interaction, drug-herb interaction, and the drug-food interaction. There is a good example about the detection of pollutants in wastewater in Thailand. Table 2 represents the concentration levels of bisphenol A and 17-beta estradiol concentration level in 5 different Thailand rivers. And figure 14 represents the HPLC UV chromatogram of separation of bisphenol A and 17-beta estradiol. Conclusion, HPLC includes separation of components with the use of the mobile phase. The mobile phase could be water, acetyl nitrite, methanol, and the stationary phase, the most used stationary phase, silica C18 or C8 column. HPLC is an effective and sensitive method for detecting pollutants in wastewater effluent, even at low concentration level. Ultra-high performance liquid chromatography operates at high pressure and with a small particle size in the column, which achieves faster and sensitive analysis compared to HPLC. HPLC detector is necessary for the quantification of polar pollutants such as pharmaceutics or female hormones in Thai rivers and wastewater influence. And here are the references for our presentation. Thank you.